Hi everyone, I'm Katie and I'll be talking about comparing methods in detecting Xylella fastidiosa. So before I start this presentation, I would like to take this opportunity to thank Dr. Lisa Ward and Dr. Suzanne and Sissy Frey for managing me and Jachi for supervising. I'd also like to thank Center for Forest Protection for the funding and the opportunity, and Forest Research for supporting me throughout the internship. So the overall aim of the experiment is to compare two protocols, one of which is being used by forest research now for detecting Xylella fastidiosa, and this involves DNA extraction and qPCR. We'll be comparing by evaluating the quality of the extracted DNA and qPCR results. And as Shachi mentioned about the importance of molecular diagnostics, we ultimately hope to improve the protocol that forest research is using. So let me talk about the general workflow or the current protocol that is being used to test samples for Xylella. First of all, we will process the sample by taking the leaf or the stem, put it into an extraction bag and add water as a buffer for homogenization. We then extract DNA and run qPCR. In the qPCR step, there are two sequences being targeted. The first one is the XF gene, which is a conserved gene in Xylella, and the second one is the 18S rRNA gene, which is a universal gene present in the rRNA of eukaryotes. And so this 18S gene is actually important in proving that there is sample DNA being sent into the qPCR and that any negative Xylella results are due to the absence of Xylella, but not simply because of the absence of the DNA. And so that's why we call that an internal control. And so if the sample has got the two target sequences, they will be amplified respectively, and the qPCR will give us a CQ value or CT value. The lower the CQ value, the earlier the sample is tested positive and the better the result. And also, I'd like to point out that we regard everything that has over 35 CQ value as negative. So to improve the protocol, we're thinking, how can we detect Xylella more effectively? Can we improve DNA concentration, or can we improve the purity of the DNA? Can the bacteria be detected more efficiently and in lower concentrations? Here in the homogenization step, we're using water as the buffer. Is there any substitutes that can better facilitate the subsequent processes that I talked about? So let me introduce you to compared to water, a buffer, a lysis buffer, used in some DNA extraction kits. It is efficient in removing contaminants or inhibitors that might affect qPCR. And so we hypothesized that samples extracted with CTAB have better DNA purity and subsequent qPCR results. So here's what we did to test this. We took leaves from an oak tree, divided them into water versus CTAP treatment groups, and inoculated them with Xylella of different concentrations. In other words, we had 6 treatments. The first one is the control, which we didn't inoculate any bacteria. Second one is we call OD1, as Jachi mentioned, and this will be the highest concentration that we inoculated. And then from OD1, we did a serial dilution, which means that we diluted the inoculum concentration to 10 to the power -1, -3, -5, or -7. So after 48 hours of incubation, we took the bottom half of the leaf with the petiole and the midrib and used water or CTAP for the homogenization. Then we extracted DNA and ran qPCR. So here are the results. In terms of DNA extraction, we looked at 3 indicators, which is first one is the DNA concentration, which is the yield. The second one is the A260 to A280 ratio, which is an indicator of the DNA purity from protein contamination. And the third one is the A260 to A230 ratio, which shows the DNA purity from salt contamination. And these were all measured by the nanodrop. So as you can see from the results, samples extracted from water with water has a significantly higher yield than those extracted with CTAP. Meanwhile, CTAP gives a higher DNA purity in terms of protein contamination, and we did not observe any significant differences in the salt contamination. And in terms of qPCR, this XF gene, we could see that negative controls did not have any positive results, which means that any positive results are due to the bacterial inoculation, which is a good thing. We're looking for that. And the second thing is samples extracted with water has more positive results in the third dilution. And lastly, the percentage of positives drops from the fifth dilution onwards for both. As for the CQ values for the same gene, only at OD1, which is the highest concentration, we observed significant differences. As for 18S, samples extracted with water has significantly lower CQ values, which means that the results were earlier. So all in all, we could see that the samples extracted with water have higher DNA concentration and earlier qPCR results in both the target sequences. Samples extracted with water also gives more positive results in the third dilution. On the other hand, CTAB is better at removing inhibitors, thus the samples extracted with it have higher DNA purity. And so putting all these together, we could see that although water is less effective in removing inhibitors, it gives more yield, and that potentially might explain the lower CQ values. Thus, we could see yield is a more important factor in giving earlier results. Furthermore, Xylella is mostly undetectable in qPCR after the fifth dilution within the plant DNA, and that forest research is using a robust protocol for Xylella fastidiosa detection. So last but not least, I would like to talk about my internship experience. I just want to say I really enjoyed my time in forest research and my internship, as I've been able to learn a lot of new skills and work with Xylella, which is a very special subject, and also gain some soft skills as well and establish connections. So thank you very much for listening.